Today I will be talking about hammerhead versus human sense of touch. Hammerheads typically live in temperate and tropical waters. Hammerheads like cooler water, therefore the reason why they are found in tropical water. They are gray-brown to olive green on the top with white underbellies. Due to these colors, it helps them camouflage well in the ocean, the tops being darker so fish and other animals can't see them from above, and white on the bottom so they blend in with the sky above the ocean. Hammerheads are a very unique animal because of its head shape. Hammerheads are the only shark with this head shape, making it more unique and more identifiable. I personally love sharks, and I am really excited to talk about them in this slideshow, specifically hammerheads. They are commonly found inshore and offshore. They can also be found over the continental shelf near islands, reefs, and in lagoons. They are commonly found in shallow water with depths of 328 feet, 100 meters, but may occasionally be found in the depths of 984 feet, which is 300 meters. I'm specifically talking about the sense of touch because the way humans feel and the way hammerheads feel are very, very different from one another. They're just different types of senses, and they serve different purposes for our lives. Our sense of touch allows us to get to know the world and people around us. We rely on tactical feedback to maintain essential behaviors like manipulating food to feed ourselves and offspring. Touch is also required for cognitive development. Touch is a part of intimacy, and it requires direct contact with the skin. Lack of touch or too much touch as a child is a hallmark of neurodevelopmental disorders such as autism disorder. Touch is mediated by mechanosensory neurons that are embedded in the skin. These neurons relay signals from the periphery to the central nervous system. Axons elongate from these neurons and make contact with the developing skin. The epithelium of the skin transforms from homogeneous tissue to heterogeneous organ that is made up of distinct layers and microdomains. Throughout this process, each neuronal terminal must form connections with an appropriate skin region to serve its function. The neurons that transform touch stimuli at the skin surface to electrical impulses, the currency of the nervous system, are known as mechanoreceptors. Two main types of mechanoreceptors innervate mammalian skin. Low threshold mechanoreceptors are turned to respond to gentle touch, whereas nigroreceptors encode force in the harmful range. Now, electroreception, the ability to detect weak, natural occurring fields. Electroreception is made possible through the existence of sensitive electroreceptor organs in the skin. Electroreception is for the detection of prey or other food sources and objects, and in some cases, communication. Electroreception was first appeared in jawless craniates, animals with skulls, such as lampreys that possess an epidermal ear and bud organ innervated by the lateral line nerve. The sensory epithelial cells resemble hair cells of a lateral line or inner ear, and the nerve fibers connecting them to the brain travel in the same nerve bundles with the lateral line nerves. The portion of the lateral line located on the head may function during feeding. Both of the ears and lateral line system detect different forms of vibration, but they are sometimes referred to together as the acoustical lateral system. As the lateral line system, the electroreceptor sensory cells connect with the outside seawater through gel-filled canals, which open at pores of the skin. These pores are located mostly around the snout, jaws, and head. Enhanced electroreceptive capabilities are unique in hammerhead morphology of hammerhead sharks. Are unique for the head morphology of hammerhead sharks. The laterally expanded head allows them to possess electric field waves much longer than other sharks. With a broader area, hammerheads have a much larger prey encounter. Comparing the behavioral response of juvenile scalloped hammerhead sharks to the similar-sized sandbar sharks that possess a typical Rakim shark head morphology. Both species were exposed to prey-simulating dipole electric fields, and their responses were recorded and quantified using digital video analysis. The sharks oriented to and a bit at the electric field as if it were a natural prey item. The hammerhead did not demonstrate greater sensitivity to the dipole electric field, but would likely demonstrate greater sensitivity if exposed to a large uniform electric field where the greater ampullary tubule length would provide an advantage. Although they were not more sensitive, the hammerhead did sample a much greater area than the sandbar sharks, with equivalent spatial resolution, thus providing a greater probability of prey encounter. And these are my sources. Thank you.